At long last, we are getting to our own kingdom, the animals. For this lecture, just a couple of learning objectives. Animals, like these iconic species you are probably familiar with. Or these, perhaps a career as a veterinarian is in your future. Or maybe just these cute and cuddly little guys. Aren't they adorable? But the fact of the matter is all these animals I just showed you are just a very small part of the total diversity of animals. Looking at this pie chart, you can see how very small our vertebrate slice is. This green slice here is for all the chordates, which includes the vertebrates. And most of that slice is made up of fishes. Perhaps a better representative image of animal diversity lies in the next picture. Well, not plankton. Most plankton are basal eukaryotes, such as we've already seen, though plankton also includes many microscopic animals and larval forms of animals. We will see all these phyla and more, though this unit will end before we get to echinoderms and chordates. So a more representative slide of animal diversity should include images such as these, all from phyla we will discuss in this unit. The largest phylum of animals is the arthropods. Maybe you remember from our very first lecture. Over 1 million known species representing more than 80% of animal diversity. And the great majority of those arthropods are of a single class, the insects, with over 850,000 species. And within that giant class, one order dominates, the beetles, with over 350,000 species. And these are just a few of the prettier ones. As I also told you back in Lecture 1, the largest family of Coleoptera, or the beetles, are the more than 86,000 species of weevils. For what it's worth, that's more than 25,000 more species of weevils than all chordates, which includes vertebrates, just for a bit of perspective. So, what is an animal, if it's so much more than just the soft and cuddly things? Animals are heterotrophic, but unlike the fungi, animals are heterotrophic by ingestion, meaning we have a special area of the body for eating, namely a mouth. Animals also display gametic meiosis. We use meiosis to produce our gametes, which are the only haploid cells. Gametes fuse to form a diploid zygote, and that zygote divides by mitosis to form a diploid multicellular organism. All animals have some type of motility, even if they become sessile or stationary for part of their lives. Most of the time, this is coordinated by muscles and nerves. And those muscles and nerves are comprised of differentiated types of cells. As all animals are truly multicellular. We may be microscopic, but even microscopic animals are made of many, many different types of cells. And another feature we find in animals that we've rarely seen in other organisms we've encountered this semester is a complete lack of cell walls. Some animals may produce shells or exoskeletons or other hardened protective structures, but these layers do not envelop individual cells. Animals do not produce cell walls. Similar to the other kingdoms, we see evolutionary trends. Here are 4 of them, which are important. Winkety wink. Planes of symmetry sounds very mathematical, but it's a pretty straightforward concept that we've seen before, as in flowers. Sponges, which are some of the most basal animals with many ancestral characteristics, are asymmetric in their construction. No planes of symmetry. If I were to make a birthday cake and you wanted to make sure everyone got exactly the same size and shaped sliced, you'd be screwed. However, if you didn't want a sponge cake, or rather a sponge-shaped cake, but preferred a jellyfish or a sea anemone cake, those are ideal for sharing because they are radially symmetrical. They are round. But the number of round animals is few compared to the number of bilaterally symmetrical animals. Bilateral symmetry should look familiar to you, especially if you've ever looked in a mirror. When animals became bilaterally symmetrical, they developed common axes or directionality. For us, as erect walkers, anterior means up and posterior means down. But for most bilaterians, anterior means forward and posterior means behind. We have a dorsum and venter, which means back and front, but for animals like this goat here, the dorsum is the back and the venter is the belly. In one phylum, we see a reversion to radial symmetry. And because this is the second time in the phylogeny of this clade that they display radial symmetry, we call it secondary radial symmetry. But that phylum lies in our future, as in after the next exam. Another evolutionary trend we see follows the complexity of multicellularity. As you should recall from about 2 minutes ago, animals are truly multicellular. It's very much our thing that we do, unlike any other kind of living thing. The sponges are in many ways simple animals, not just in their asymmetry, but also in having only a cellular level of organization. They have a number of different types of cells, which we will draw, but they function as collections of different types of cells without any tissues or organs. In the radially symmetrical animals, we do encounter tissues where different types of cells work together towards organism-level purposes. Mouths, which are essentially just openings that lead to guts, which can then channel nutrients to the rest of the animal. If we can coordinate different types of tissues into more complex structures, then we have organs, and the organs can be coordinated into organ systems. Perhaps you've never thought of a lowly snail or worm as being sophisticated. If you take one apart, they're very difficult to reassemble. They have many needs associated with the intricacies of being larger, living on land, finding mates, avoiding predators, and eating. And this requires organs and organ systems to maintain. Where do all those different types of cells and tissues and organ systems come from? The instructions come from the genome. Where does the genome come from? The entire genome is present in every new cell in an organism, starting with the zygote. The zygote divides by mitosis and ... forms a multicellular organism. But what takes place in that ... Development takes place. Differentiation of cells into different cell types. Otherwise, we'd just be 37 trillion totipotent zygote cells, and that's not a look. And while most zygotes look the same, animals look vastly different to each other, and the major differences get sorted out as embryos develop. There are certain patterns to this, which we will draw, which display certain evolutionary trends. There are some clues, as well as important terms to know, in this slide. Many animals have body cavities, spaces inside to accommodate the various tissues, organs, and organ systems. But not all of them do. In the most ancestral lineages of animals, the only cavity inside is the lumen of the gut. These animals are constrained to be either very small or very much dorsoventrally compressed, i.e. flat, in order to allow for respiration. Then, as animals developed organs and organ systems, they required a space to put those organs instead of being embedded within the muscles. So the first spaces for organ accommodation evolved. And even though it evolved first, it was discovered second, so it was saddled with the pseudo tag of pseudocoelom. In pseudocoelomates, there is a body cavity to hold organs, but the body cavity is not lined completely with tissue derived from the mesoderm. That lining with mesoderm is the next step in the process, and the most derived condition is to have a body cavity lined with mesoderm-derived cells, also known as the coelom. Here we see an inferred phylogeny of the animals. The porifera, which is the phylum of sponges, are shown as the earliest diverging animals that are still living. And not surprising, given how they have no planes of symmetry and only a cellular level of organization. With the evolution of true tissues and radial symmetry, we enter what have been called the true animals, the eumetazoa. And when bilateral symmetry came along, a third germ layer was added as mesoderm. This concludes our first foray into our own kingdom and a recap of the learning objectives. And next we begin our journey into the diversity of the animals, and we start with the least animal-y of the animals, the sponges.